And so I'm going to keep the introduction really short. So basically, I'm trying to use cardiac regeneration as a way to find a treatment/cure towards heart failure, which is a very common clinical condition. So a brief introduction. So the mammalian neonatal mouse heart is able to regenerate within a week. So if you surgically apically resect the heart at P1, the heart will completely regenerate within 21 days. And this is primarily mediated by the proliferation of pre-existing cardiomyocytes. However, this remarkable ability to regenerate is quickly lost by postnatal day 7 or P7, and this coincides with cardiomyocyte cell cycle arrest. And so the reason for this is there are a number of reasons for why this happens. There's a number of transcription factors that decrease, and a lot of the cell cycle genes are decreased as well. And we think this is mediated by the difference in environment. So in the in utero environment, the oxygen levels are very low. And postnatally, after the mouse is born, it's exposed to a highly oxygenated environment. So it transitions from an anaerobic glycolysis approach to a mitochondrial oxidative phosphorylation approach to deriving energy. And so this it particularly focuses postnatally in using fatty acid utilization, and this approach of using fatty acid utilization is energetically favorable. But the problem is that the mitochondria produce in the cardiomyocytes produce more reactive oxygen species, and this stimulates the DNA damage response, which subsequently leads to a cell cycle arrest in most cardiomyocytes. So from after P7, most cardiomyocytes rarely divide in the body, and that's why when you resect the heart at P7 onwards, the heart no longer regenerates. So we predict that this switch in energy substrate utilization is also responsible for the decrease in cardiomyocyte cell cycle progression. So we therefore ask the question, can we inhibit fatty acid utilization to promote cardiomyocyte proliferation in the postnatal mouse heart? We have two main approaches, which I'll go through. One is a dietary model and one is a genetic model. Our first aim is to determine the effect of depleting dietary fatty acid intake during the lactation period and its effect on cardiomyocyte proliferation. The second approach will genetically try to increase glucose relative to fatty acid oxidation by promoting pyruvate dehydrogenase kinase by deleting PTK4, which is pyruvate dehydrogenase kinase 4. It's the main isoform found in cardiomyocytes, and its role inhibits PDH in an environment where there's a lot of fatty acid in the presence of a high level of fatty acids. So we hope to, this approach will hopefully decrease the reliance, will promote glucose utilization at the expense of decreasing fatty acid utilization and decreasing reactive oxygen species. So yeah, so the hypothesis is that if we stimulate glucose oxidation and decrease beta oxidation, will decrease the production of mitochondrial reactive oxygen species and therefore decrease the DNA damage response and hopefully promote cardiomyocyte proliferation. Okay, so we'll begin. So we're going to begin with the dietary model. And so this is just a little bit confusing. So basically, we're using a double transgenic mouse to generate a mother that would produce milk that is deficient in fatty acids. And so to achieve that, we use a flox-perk mice crossed with a mammary gland-specific MMTV cream line. And this will generate a double transgenic mouse that has a deficiency in PERK in the mammary glands. And what that does is that this deficiency in PERK and mammary glands inhibits key lipogenic enzymes, which subsequently ends up with a production of milk that is deficient in fatty acids. And there's more information in this reference, which you'll find in the paper that I published for this project. And so these pups are born and they are exposed to a fat-deficient milk from the mother. And by P12, when the pups begin to nibble food, we introduce a fat-free diet. And these hearts are collected at P21 and 10 weeks of age. And for the controls, we have the reverse situation. We have the father that is the double transgenic. The father doesn't make milk, so that doesn't matter. And then the mother is just the flox PERK by itself. So all the pups are genotypically similar between the fat-deficient milk group and the control milk. So the next series of slides will address the P21 time point for the fat-deficient milk model. And so what we see from the fat-deficient milk, from the mice exposed to fat-deficient milk at P21, the heart weight to body weight ratio is increased compared to the mice from the control milk. And this was also associated with a decrease in cardiomyocyte cell size in fat-deficient milk compared to the control group, as assessed by wheat germ agglutinin staining. So in addition, the fat-deficient milk group seemed to promote cardiomyocyte proliferation, as assessed by PH3, which is phosphorylated histone H3 at serine 10. It's a mitosis marker. And we see an increase in PH3 positive cardiomyocytes in the fat-deficient milk versus the control. To assess for cytokinesis, we looked at Aurora B kinase. And so in the fat-deficient milk, we see an increase in Aurora B positive cardiomyocytes compared to the control. We also isolated the total number of cardiomyocytes and counted that. And we see a total increase in total cardiomyocytes in the fat-deficient milk group compared to the control. So next, because as many of us in the field know that at P4 in the mouse heart, binucleation occurs. So basically, cardiomyocytes undergo karyokinesis, DNA synthesis, but without cytokinesis, mostly anyway. And so we use this interesting mouse model called MANAM. It stands for Mosaic Analysis with Double Markers. And we crossed it with the alpha MHC mercury mer mice. So I'll try and explain this briefly, but if you want more details, it's in this reference, Ali et al., which is embedded within the reference section of my paper. So the MANAM model itself is a double transgenic mouse. It consists of two alleles. It has, the first half is a green fluorescent protein nucleotide sequence, followed by the second half of the red fluorescent protein sequence. And the second allele has opposite arrangement, the first half of RFP and second half of GFP. Both of these alleles do not produce any colored protein. And so after S phase and DNA replication, and we inject the mice with tamoxifen at P7, the Krelux recombination will generate a full-length GFP and a full-length RFP, as a nucleotide sequence, and it'll produce GFP and RFP. So in most cases, the cells will not, the cardiomyocytes will not undergo cytokinesis and will form a cell that contains, cardiomyocyte that contains both GFP and RFP, and it would appear yellow, like in this image down here. And with a smaller proportion of the cells, cardiomyocytes will undergo complete cytokinesis, and you'll get cardiomyocytes that are exclusively red or exclusively green, like here and here. And so to calculate the cardiomyocyte renewal rate, this lineage tracing model we can address that by calculating the number of single labeled cells, like red divided by the number of yellow cells. And so here, the control mice, we get roughly a 1% cardiomyocyte renewal rate compared to the fat-deficient milk group, which has roughly 1.5%. And in the green cells, it's also the same percentages. So we expect the number of single label cells in the control group for green and for red to be similar, and also the fat-deficient milk for green and red to be similar. It's kind of an internal control. You expect the proportion proportions to be the same. So basically, this proves that we are getting newly generated cardiomyocytes from the fat-deficient milk group. Okay, so next we looked at the 10-week time point. So the next set of slides will be in reference to the 10-week time point. At 10 weeks, the fat-free diet didn't have any difference between the heart weight to body weight ratio. But however, the cardiomyocyte cell size was smaller in the fat-deficient milk group. We also assessed the number of Ph3 positive cardiomyocytes, but there were no difference between the two groups. The data is not shown on this slide. We also performed a lipidomics profile by mass spectrometry to assess the lipid content in the hearts of fat-free diet versus the control. And all the components are similar, with the exception of phosphatidylcholine, which is decreased in the fat-free diet. Next, we looked at the liver with the same experiment, and every component was similar between the two groups, with the exception of triacylglycerol being high in the livers of the fat-deficient milk. And we saw an increase in the liver weight to body weight ratio in the fat-free diet. And you can see from this image, the liver is much larger in the fat-free diet. And in addition, as revealed by H&E staining and oil red O staining, we see a lot of fat vacuoles in the liver, and this is consistent with a phenotype for liver steatosis. So in summary, for the first half of the talk, the fat-deficient milk/diet prolongs the cardiomyocyte proliferation window, but not until adulthood. And the fat-deficient milk/diet leads to a compensatory increase in the fatty acid synthesis in the liver, and this results in liver steatosis and also a cessation of the early cardiomyocyte proliferation that we observe at P21. So by the adult stage, we don't see the cardiomyocyte proliferation anymore, likely because the liver now is producing fatty acid for the body. The second half of the talk, we're going to address the genetic model of promoting glucose utilization at the expense of fatty acid utilization. So mitochondrial PTH is a critical enzyme for converting pyruvate into acyl-CoA, and PTK4 is the main isoform in cardiomyocytes. So our strategy is to delete PTK4 to promote pyruvate dehydrogenase to allow this pathway for the glucose utilization to generate ATP and hopefully decrease the reliance on fatty acid utilization. Okay, so to do this, to generate a cardiomyocyte-specific deletion of PTK4, we made a double transgenic mouse. We crossed a PTK4 flox mice with a cardiomyocyte-specific alpha-MHC mercury mice. And so these mice and the control mice were, in the adult, were injected with tamoxifen and collected 2 weeks later. We see in the Western blot here that in the control mice, the PTK4 band is very strong. And in the inducible PTK4 line, we see that PTK4 has been successfully deleted. And as you can see from this little diagram here, PTK4 inhibits PTH. So if PTK4 is truly deleted, we expect the PTH activity to increase. And so in this graph here, we do indeed see an increase in PTH activity in the PTK4 knockout, inducible knockout mouse. So next, to determine the rate of glucose and fatty acid oxidation, we use carbon-13 NMR, nuclear magnetic resonance, on these isolated hearts. This experiment was performed by collaborators of ours, but in short, what they do is they isolate the heart and put it on a Langendorf-like system, and they feed in carbon-13 labeled glucose or carbon-13 labeled long-chain fatty acids, one at a time, but not together. And based on the system, it generates a readout of different carbon numbers on the glutamate isotopinase. And based on the carbon number that is oxidized, we're able to retrospectively find out work out the proportion of glucose and long-chain fatty acid that has been oxidized. And so with that, it generates this graph. So the diomyocyte-specific deletion of PTK4 results in an increase in carbohydrate fractional oxidation and a decrease in long-chain fatty acid fractional oxidation. We also see an increase in PDH flux in the PTK4 knockout, as well as an increase in the TCA flux. So these results show that, that PTK4 plays an essential role in the control of PDH activity and substrate utilization in cardiac muscle. Next, we examine the effects of the decrease in fatty acid oxidation from the PTK4 deletion on heart size and cardiomyocyte size. So the heart weight to body weight ratio was not different between the two groups, between the PTK4 inducible knockout and the control, but we did see a decrease in cardiomyocyte cell size. And this was associated with an increase in cardiomyocyte proliferation. We saw in the PTK4 inducible knockout, we saw an increase in Ph3 positive cardiomyocytes and also an increase in cytokinesis as assessed by the increase in Aurora B positive cardiomyocytes. We also saw an increase in total cardiomyocytes in the PTK4 inducible knockout. Okay, so as I mentioned before, our working hypothesis was that if we promote glucose utilization and decrease fatty acid utilization, we'll expect to see less ROS production and probably a decrease in the DNA damage response. So this is in fact the case. So, so on the left panel here, we are looking at oxidative stress and DNA damage as assessed by 8-hydroxyguanosine. In the control, you can see this cardiomyocyte nucleus has a lot of 8-hydroxy nuclear foci compared to the bottom panel here in the PTK4 inducible knockout. The cardiomyocyte has much less. And this is shown graphically right here. And then we looked at the DNA double-stranded breaks by gamma H2X. And again, in the control, you see a lot of nuclear foci in the control compared to the PTK4 knockout, which has a lot less. And lastly, to assess for the DNA damage response, we looked at phospho-ATM. And again, in the control, there's a lot of nuclear foci in the cardiomyocytes compared to the PTK4 inducible knockout. And graphically shown, there's a lot less as well. So taken together, all these results show that by inhibiting PTK4, this promotes glucose utilization, decreases fatty acid utilization, and decreases the DNA damage response, and subsequently leads to an increase in cardiomyocyte proliferation. So we also performed RNA-seq on the PTK4 hearts versus the control, and about 270 genes were downregulated, including P21 and P57. 420 genes were upregulated, including cell cycle activators. And for the gene ontology, we saw all these changes. I'm running, I think I'm over time, so I'll just quickly finish off. So next we assessed the PTK4 inducible knockout model in a setting of myocardial infarction. We did a permanent ligation of the left anterior descending coronary artery, and at 1 week post-MI, we performed echo and also every month until the 6-month time point. We injected tamoxifen to delete PTK4 at the 1 week post-MI time point. Basically, if looking here, this is the 1 week post-MI echocardiogram in the control versus the PTK4 knockout. They're similar, but at 6 months post-MI, the control heart looks really bad compared to the PTK4 inducible knockout, where the heart function looks much better. Looking at the ejection fraction at every time point, this is before MI, this is 1 week post-MI. And if you follow the blue curve, this is the control where the ejection fraction continues to decline with time, compared to the PTK4 inducible knockout, where this prevents the decline in ejection fraction. And by 6 months post-MI, it actually improves the ejection fraction compared to when it started the tamoxifen injection. Lastly, the left ventricular dilatation was less in the PTK4 knockout compared to the controls. The infarct size was smaller in the PTK4 knockouts, and also the cardiomyocytes were smaller in a post-MI setting in the PTK4 knockout compared to the control. There was also an increase in cardiomyocyte proliferation in the PTK4 knockout in the setting of post-MI for Ph3 positive myocytes, and an increase in Aurora B positive cardiomyocytes. So in summary, fat-deficient milk prolongs the cardiomyocyte proliferation window, but not into adult hearts, by decreasing fatty acid utilization by way of deleting PTK4 to promote glucose utilization, and this decreases the DNA damage response and promotes cardiomyocyte proliferation. After myocardial infarction, the cardiomyocyte-specific deletion of PTK4 improves the left ventricular ejection fraction and decreases the left ventricular dilatation and remodeling. I'd like to thank all these people, especially my co-first authors, Alison Cardoso, Jenny Savler, and our mentor, Hesham Sadek, who's a really great scientist and mentor, and it's a real privilege to be able to work with him. The main funding bodies for this project is NIH and AHA, and I'll just put up this paper that was recently published. I want to thank all the co-authors that are listed here, and I'm going to apologize in advance that I can't answer all your questions in the time frame, so you'll most likely find the answers in this paper. So with that, I'll be happy to take questions. Thank you.
Great, thank you, Nick. Well done. And so we've got a few questions, about 5 minutes for questions, so feel free to post them on the Q&A section, and we'll try to get around to them. So one of the questions here is, do you have any data about what happens to glycolysis in the PTK4 knockout hearts?
Well, what do you mean? Like, because like I showed the fractional, it increases in that that carbon-13 NMR experiment. So I guess it increases. Is the answer to that question?
I mean, I guess I'm assuming sort of looking at normal homeostatic glycolysis relative to what is going on in the PTK4 knockout. Are we looking at something that could be potentially, super physiological activity there, or potentially having
that experiment controlling? I mean, that was compared to a control heart, so I guess like, I mean, I don't know how unphysiological that level is, but yeah, it's increased. Yeah.
Okay. Another question in regard to the mechanism by which glucose, moving around here. Here we go. Increased glucose utilization leads to ROS production in cardiomyocytes. Is that mechanism, the link between ROS production and glycolysis, known?
Actually, where is the text for the question?
So if you just open up the Q&A.
Are you doing this chronologically, or whose question is this? The name of the
this is Kalia Primer.
Okay, thank you for presenting. It's a mechanism by which increased glucose utilization leads to. I'm not sure how to answer that question, so I'm going to pass.
Okay, no worries. In regard to, this is from Yao Keet Sam, could you speculate on the changes in the phosphatidylcholine in the hearts of the fat-free fed mice?
Okay, so if you speculate on changes, the fat-free, I'm not really sure how to answer that question. I'm actually more of a cardiac regeneration person than the metabolism aspect, and all the questions seem to be about metabolism.
All right, so let's see if we can find some more specifically around the regenerative biology here. I was actually, I mean, maybe you could answer my question here in regard to the timeline after the PTK4, when you just acutely add the tamoxifen, how long does it take to actually see the effects of the myocyte size, after you start to change over the biology around the energy substrate utilization?
Actually, one of the questions that one of the reviewers did ask, how long does it take for PTK4 to decrease? And so it actually, we did an experiment where we did 3 injections of tamoxifen, 1, 3, and 5 days. By the 4th day, PTK4 was already completely abolished. And the half-life of PTK4 was only about 1 hour. So the deletion of PTK4 is early. Normally when we look at the cardiomyocyte cell size, we kind of waited a little longer because these changes take a while. The basis of the decrease in cell size is speculated to be an increase in cardiomyocyte proliferation. So we think that when we get more newly generated myocytes, we'll decrease the average cell size. So for us, we waited like 3 weeks, but it could be earlier, but we decided to pick P21 as earliest time.
Another question that's related to this, again, going to the fat-free milk model that you generated there, is there an effect beyond the liver and the heart in regard to normal development? Because I would imagine there's likely to be some substantial impacts in terms of normal organismal biology. I mean, one of the questions here is in regard to the body weight of the mice that were fed on the fat-free milk that may be implicated or sort of secondary effects in regard to cardiac biology there.
Actually, I didn't show the figure, but in figure 1C, there's actually no difference in body weight. Between the two groups, surprisingly, the body weight is similar. I mean, the reason why we chose the liver was because when we opened up the chest to dissect the mouse, it was very obvious that there's this liver that's just like in your face, basically, and it was quite pale, and that seemed to suggest it was quite fatty. We didn't look at other organs, but there could be an effect on other organs too.
Okay, so I've entitled my talk "Cardiac Repair Using Recombinant PDGF and Pluripotent Stem Cells," and the reasons for that will become clear. This introductory slide's almost redundant, given the audience. If you have tuned into this webinar, you'll know that we're trying to go for regeneration of the heart to restore function to hearts that have lost it in a way that drug therapy currently just can't do. And that heart transplantation and left ventricular assist devices, although have been a very important advance, they have their significant limitations. A lot of my work looks at heart failure or at least heart dysfunction after myocardial infarction. And that's primarily because of the animal models that we use. And we hear a little bit that myocardial infarction has been done and we need to look away from this. But I point your attention to this echocardiogram here of a patient of mine a few years ago now. And he came in with a myocardial infarct on the background of a smaller one. So he came with a large anterior infarct. And despite being able to reperfuse his coronary arteries, he died of overwhelming heart failure 3 days into his admission. And in my mind, and to most clinicians, a myocardial infarction is not done. So cardiac repair and regeneration really got started, I suppose, with the concept of stem cells. And now we have a number of different modalities that we're investigating. But I think it's important to just point out, for those who might not be as entrenched in the field, the difference between the types of stem cells. And my work, as you'll hear shortly, has encompassed both of these. So this cartoon, I like it. It's old now, from 2005, but it shows this necessary hierarchy, going from the totipotent stem cells and the pluripotent stem cells to adult stem cells. And adult stem cells having more restricted differentiation potential that usually is surrounded around the organ system from which they're found. Of course, induced pluripotent stem cell technology now has blurred those boundaries a little, and we can now reprogram adult cells to become like embryonic stem cells. But it's useful to keep that in mind and to bear the differences between the two. And the field has not always paid attention to that. And this was the case put forward in this review published in Nature Biotech last year. Now, this is looking at papers. Well, this figure here is looking at papers in favor of the presence of adult endogenous cardiac stem cells, and they're in blue at the top. And then papers published against the existence of endogenous cardiac stem cells. And by a cardiac stem cell, they're talking about a universal cardiac stem cell, a cell residing within the adult heart that can give rise to all lineages within the heart. And in particular, the sticking point was cardiomyocytes. Anyway, it's a good read. I'd recommend it. And it's interesting to reflect how this timeline shapes the research around us, and specifically for mine. So as Nathan mentioned, I started my PhD with Richard Harvey here in 2007 on the back of all of these publications. And in fact, that's why I entered this field, because of this wonderful possible cardiac stem cell. And I went and chose a postdoc in a lab that studied pluripotent stem cells, largely because of the inability of some of that differentiation potential of those cells. So the first part of my story looks at a growth factor, PDGFAB, and the effects that we've seen in restoring cardiac function after myocardial infarction. And this story started with my PhD with Richard, and we characterized cells found in the adult mouse heart that we felt had mesenchymal stem cell-like properties. And the cell surface signature contained the tyrosine kinase platelet-derived growth factor receptor alpha. Now, it's an important differentiating point to say that not all of those stem cell-like cells express, this signature did not define all of this cell population, but the stem cell-like cells existed within the cell population that had this signature. And my contribution to this quite extensive body of work was predominantly looking at the lineage origins of these cells. And I had a number of different Crelox lineage tracing mice, and I concluded that these cells were derived from the developing proepicardial organ and not from the bone marrow. I won't go into the details, but those aware of cardiac embryology will know that most of the cardiac fibroblasts, if not all of the cardiac fibroblasts, are derived from this proepicardial organ. And then Richard's group went on to explore the possibility of being able to stimulate these stem cell-like cells without the need to isolate them, which was quite different to the many groups that were trying different cell therapies at the time. And he was able to show that fractional area change, that is a parameter of left ventricular function, improved when he delivered systemically PDGF compared to the vehicle only. And that's seen best in the apex, but also as you progress up in the left ventricle more basally. So we now had the hypothesis that PDGFAB would stimulate cardiac fibroblasts and likely within that some MSC-like population. And from what we knew about these cells, we felt that it would impact vascular formation and collagen deposition and matrix within the heart. And so it was on that background that a cardiologist who started a PhD with me, Sujitha Dava-Palachandran, set out to set up a preclinical large animal model to test these findings. And this was, as Nathan mentioned, published earlier in this year. So you can look at the details there, but I'll just go through it quickly here today. 3 groups, infarct and a sham group, but those that received infarction were randomized to have either vehicle alone or the recombinant protein PDGFAB. And the baseline cardiac magnetic resonance imaging was to assess the infarct size and then a number of endpoints, which I'll go through in this talk. So we delivered the recombinant factor by osmotic mini pump, and we showed that the factor PDGF, which does occur endogenously, was elevated above those endogenous levels by ELISA. And then here, this graph here is showing that late gadolinium enhancement, which is a gold standard surrogate for scar after infarction in clinical cardiac MRI. So the scar was not significantly different between the vehicle and the PDGF treated animals. In contrast, the left ventricular ejection fraction, how well the left ventricle pumped after infarction, changed quite significantly. You can see in the delta on the right of screen. And so the PDGF animals, 28 days after infarction, improved their function almost towards the sham levels. So this was quite a robust finding. And with cardiac MRI, we can look at the volumes as well. And you can see that there was a significant difference in the left ventricular end systolic volumes in 28 days compared to baseline. And the diastolic volumes did not change in a statistically significant manner. Now, this is important because a lot of left ventricular functional change in most therapies is driven by changes in diastolic volumes. And it's generally felt that diastolic volumes are an earlier indicator of improving or declining ventricular function. So this was surprising and of interest to me in particular, that we're affecting systole more so. So we were also able to do micromanometer studies, and here analyzing the change in pressure and over time in systole and diastole just confirmed our MRI findings of functional improvement. And the beauty of these pressure volume loops, which were performed with Michael Fennelly and Scott Kesterman, is that we can create load independent parameters. So most people who are aware of cardiac functional models will know that changes in preload and volume and beat to beat variation can affect your results very significantly. So in this model, we can occlude the vena cava and shrink the cardiac output to almost nothing. And in doing that and creating these pressure volume loops, we create load independent measures. So that's the end systolic pressure volume relationship, which was very significantly different in the factor treated versus the vehicle treated groups, as well as the area under the curve, which is the stroke work. And these are load independent differences. So all of that's telling us we have very significant functional change in these animals. But what was causing the change? And that's where Rob and Kalyan in our group looked at vascular formation. So this is from Willebrand factor staining in 2D sections first, quantified, showing more vessels in the infarct region of PDGF-treated animals compared to control. But the beauty of these studies is that Rob and Kalyan went on to assess this in 3 dimensions, three-dimensional tissue, using cleared specimens, using the cubic R3 protocol. And here's some of the analysis of that. This is smooth muscle actin stained 3-dimensional tissue and analyzed using this program known as IMARIS. And you can see the vessels that are formed in the PDGF treated animals is very different to those in vehicle treated animals. And I'll show you the quantification here. So we're seeing more dense vessels. We're also seeing more branching points in PDGF and interestingly small vessels. So our thinking is that there was more vessel sprouting or possibly, and we're unable to prove this, more new vessels being created after PDGF treatment. So I showed you the late gadolinium enhancement at day 2 as a surrogate for scar. Now this is at day 28, and the graph, you might recall, is looking very similar to the day 2. And that is to say that there was no statistically significant difference in the scar. Now, and this was quantified by histologically by Gamori trichrome staining as well. And this surprised us again, because I've already shown you that we had a robust functional change, and usually people would think that would go hand in hand with less scar. but that's not what we were seeing. So we started thinking, well, maybe if it's not the quantity of scar, it might be the quality or something about the scar itself. So Rob and Kalyan looked at the collagen fibers by second harmonic generation 2-photon imaging, or a polarized light birefringence as well. And hopefully you can see here that in the vehicle, they're less organized, and after PDGF treatment, we're getting a lot more alignment of the collagen fibers. And that's what Rob found when he went on to quantify this. So you can see after PDGF treatment, a lot more alignment of the fibers. The absolute value does not matter. It's just when they're grouped together, the angles are aligned. When cardiologists think about scar, we think about dangerous arrhythmias, and we performed programmed electrical stimulation. That's a way of assessing which infarct patients might go on to have dangerous arrhythmias. And it was a lot harder to induce these dangerous arrhythmias in the PDGF-treated animals compared to the vehicle. And when we analysed all the death after procedural-related mortality was removed, we found that all of the death occurred in the vehicle-treated and not the PDGF-treated animals, which was interesting to us. So to summarise, we feel that PDGF, administered systemically in this case, can stimulate cardiac fibroblasts or a mesenchymal stem cell population within that group to influence collagen alignment, scar formation, and vasculogenesis within the infarct region. And the outcome of this is improved ventricular function, decreased arrhythmias, and death, which is obviously very important to our infarct patients. So I'll shift gears now and talk to another major theme within our group's research, and that's looking at human pluripotent stem cell-derived cardiomyocytes. These videos, I'm sure everyone has seen. So these are cardiomyocytes that are generated in the lab from pluripotent stem cells, either embryonic or induced pluripotent stem cell-derived. And suffice to say, you get robust functional cardiomyocytes, and we grow them in the group by the millions, literally. And it's an exciting time in the field. Just the other week in Nature News, we found out that 2 patients in China have been treated with induced pluripotent stem cell-derived cardiomyocytes, and that there are patients in Japan that also have been treated with this technology, although that is in the Japanese case with engineered constructs. All of that work is not published. All of the details are not released, but apparently the patients are doing well so far. On the back of this, there's at least another 4 or 5 groups in North America and Europe looking towards a clinical trial, either with cell injection or with an engineered heart tissue. And they're all going to come out within the next few years, so an exciting time. Now, as Nathan mentioned, when I was with Chuck Murray, we developed a monkey model of heart dysfunction and cell transplantation. In fact, Nathan was the one that genetically altered these pluripotent stem cells to express the reporter. And this study really showed clinical feasibility of this method. Up until then, no one had really generated clinical scale myocytes from this technology or shown its efficacy or its possibility in a large animal model. And since then, Chuck's group has again published more results confirming our initial findings. And Michael Flam, who's now in Toronto, has also published similar large animal results. But the point here is that there are major hurdles to clinical translation still, and that is ventricular rhythm abnormalities that occur after transplantation and the poor cell engraftment. So, I mean, in the initial studies, we injected 1 billion cardiomyocytes, and most people are decreasing that. We're using 750 million now, but that's still a lot, and the minority of these cells actually engraft in the hostile environment. So I'm not going to show a lot of data here because, well, largely because the model's taken quite a while to get up and running. But this has been led by a cardiologist, Dinesh Selvakumar, who's now doing his PhD with me, and also Zoe Clayton, basic science postdoc with her group. And we're doing this in a pig model. And you can see that this pig has a jacket on because they're immunosuppressed intravenously. So that's protecting that Hickman's line. We have a very nice setup where we can perform human-grade angiography and electrophysiology studies, and we perform cardiac MRI here. So the table's just showing why we have chosen this pig model. We also do sheep here at Westmead, but and that is that the pig heart better resembles adult hearts in size and heart rate compared to rodents, but also small macaques as well. Like Chuck and Mike and what I found earlier in Chuck's lab, we do find ventricular rhythm abnormalities in the injected animals, and that's what you're seeing here by telemetric analysis. And we've, both Chuck and Mike have shown this as well, but we performed our own complex electrophysiological mapping to find out where these rhythm abnormalities are coming from. So this is a rendition of the left ventricle, and it's showing a heat map showing the wavefront of depolarization across the left ventricle. And here is a video of that during one of these ventricular arrhythmias that occurred spontaneously in this animal. You can see it originating from this precise location on the inferior aspect of the left ventricle. So a major difference, I suppose, from our group's work is that we feel the input cells really need to be characterized better. And that's where Zoe, and with some help from Laurentius here, have developed a high-dimensional flow cytometry platform that can analyze 12 cell surface markers simultaneously and sort those cells as well based on that profile. Now, these Tisney graphs here are showing the different cell populations, and I won't go into the details of that. So just to summarize my talk, I feel that PDGF-AB is a promising therapy for activating mesenchymal cells and possibly progenitor cells within the infarcted heart, and pluripotent stem cell-derived cardiomyocytes also remain a promising therapy. And our approach is really to better characterize the input populations to solve unaddressed hurdles. I've thanked most of my group who've been responsible for the work as I've gone along, and I think I've mentioned most of the collaborators as well, apart from Peter Gray and Andrew Prowse at UQ, who are now involved in the pluripotent stem cell-derived cardiomyocyte project, adding a tissue engineering bioreactor approach. I might also just in passing mention that from time to time we have a PhD and postdoc opportunity. So if this work interests people, please feel free to contact me. So I'll just stop that talk there and open to questions.
Great, thanks, James. So post any questions you have on the Q&A spot there on the channel. Just maybe I'll start out. I was interested in your diastolic dysfunction, or at least no substantial improvement in diastolic performance with the PDGF. I was also noting that while MRI didn't really show anything, that the in vivo hemodynamics had a significant increase in the negative dP/dt. Yeah. So it seems there is probably some diastolic benefit. And I wanted to see if you elaborated, or have you had a chance to look at that through like Doppler or any other measure? Yeah, so there was definitely changes in diastolic function. That's clear. So in the MRI volumes, it did not reach statistical significance, but there still was a trend. And in the dP/dt, there was a statistically significant difference. The pressure volume loop tau measurements on load-independent measures did not also reach statistical significance. So it was there, but to a lesser extent than systole. Mm-hmm. So, and then I suppose you were asking me what's causing that. And in our mind, it's really coming down to the matrix. We haven't exactly pinpointed why systole is impacted more, but we think it's got something to do with the collagen alignment and the changes in the matrix. On that point, I was noting on the deaths in regard to the survival curve, most of the deaths in your control were very early on.
Yeah.
And I'm wondering if you can elaborate a bit more on that, because it would seem to me that the benefit is largely in the long-term remodeling.
Well, we don't know that. So you're right to pick up on that. And the programmed electrical stimulation was late. So that was prior to endpoint. So there's probably two things that are happening with arrhythmic deaths in these animals. Something happening early and PDGF treatment. So all of those early deaths were occurring in the vehicle animals when PDGF would still be eluting by the osmotic mini pump. So it's our feeling that PDGF is acting on possibly the inflammatory system in the post-infarct environment, or doing something early that is affecting early death. And that's different to the late re-entrant scar-mediated arrhythmias. But these are all hypotheses. So we've got another PhD student who's looking at large animal work to nail down that mechanistically a little more.
Great. Still not seeing any questions on the channel, so I'll just keep firing them away. Anybody can post as you need. In regard to the stem cell characterization, I guess the hypothesis here is that there's somehow a subset of pluripotent stem cell derived cardiomyocytes that are likely to be more effective or engraft more efficiently into the infarcted heart. I guess you're trying to use these cell surface markers to parse out those populations, and then do you have any preliminary data to indicate that there's some specific bias in regard to what cell types are most beneficial?
So not in a cell therapy environment. We do have some preliminary analysis showing some interesting populations we're focusing on, but we haven't taken that into the cell therapy area. Mm-hmm. Another avenue that we're also pursuing is teasing out the different cell populations that could be used for disease modeling. So i.e. Pulling out cells that are more atrial phenotype or more pacemaker or nodal phenotype. Mm-hmm. So we feel that that'll also be of use.
Mm-hmm. Absolutely. One quick question here in regard to where you placed the osmotic mini pumps for the studies in the pig.
Yeah, so they were infused into the internal jugular vein.
Mm-hmm. Let's see here. I think that, here we go. Can we inject the PDGF right at the MI region? Have you tried any direct injections as opposed to the infusion?
Yeah, that's a great question. So we have an ongoing study conjugating the protein to nanoparticles. That's in collaboration with Steve Weiss. That's still ongoing. We don't have all the data, so that's one approach. We've also considered infusing it directly down the coronary arteries, but we haven't actually gone ahead and done that experiment. PDGF has a very short half-life, so I'm not sure intracoronary infusion would be best, but the nanoparticle approach might work, where it will hang around in the infarcted myocardium longer.